Hello everyone, my name is Min Zhang. Today, I'm going to present cadmium and indium phosphide quantum dots impacts on ML-1 thyroid and HeLa cells and their inner traffics. The overall mortality rate of cancer patients remains high. There are still some doubts about the mortality of cancer and the effectiveness and side effects of conventional treatment. Quantum dots has provided new insights into the treatment of cancer. Although quantum dots are useful in many biomedical applications, their impacts on human cells are largely dependent on the size of and functional groups attached to quantum dots, as well as the types of cells used. To address the potential harmful effects and transit routes of red and green cadmium and indium phosphide quantum dots, we explored late-stage ML-1 thyroid cancer and HeLa cells by measuring levels of cell viability, oxidative stress, and apoptosis. We hypothesized cadmium and indium phosphide quantum dots will have more toxic effects on HeLa than ML-1 cells, showing through decreased viability, increased ROS, and increased apoptosis. After we treat ML-1 thyroid and HeLa cells with 4 different quantum dots, cadmium green, red, indium phosphide green and red, the result showed there's no cell viability reduction of ML-1 thyroid cancer cells. Yet, HeLa cell viabilities were reduced by cadmium green, indium phosphide green, and red quantum dots. We further tested the reactive oxygen species, ROS, and apoptosis of ML-1 thyroid and HeLa cells. And the result indicated there is no superoxide stress produced in ML-1 cells, while cadmium red quantum dots cause an increase of superoxide stress on HeLa cells. All quantum dots cause late apoptosis of ML-1 and HeLa cells. Moreover, the toxic effects of those 4 quantum dots on non-cancerous cells, fibroblast cells, was tested to see if they can cause significant normal cell damages. The result showed indium phosphide quantum dots cause cell viability reduction of non-cancerous cells, but it is not significant since standard deviation is pretty low. As a result, ML-1 is less sensitive to cadmium and indium phosphide quantum dots compared to HeLa cell since there's no cell viability reduction. HeLa viability was reduced mainly by late apoptosis. Also, there's a very low toxicity of cadmium quantum dots in non-cancerous cells. Indium phosphide quantum dots is not harmful to non-cancerous cells at all. Since HeLa and ML-1 thyroid cells show different sensitivities to cadmium and indium phosphide quantum dots, it is necessary to study the uptake dynamics, transit routes, localization, and excretion of quantum dots in both ML-1 and HeLa cells. We hypothesized HeLa cells take up quantum dots quicker than ML-1 cells, and the intracellular localization of quantum dots is endosome. At present, little is known about the uptake and excretion dynamics and the internalization pathway of quantum dots in cancer and healthy cells. If we can fully understand the cellular uptake, targeting, and intracellular transport of quantum dots, we can optimize and enhance the safety by adjusting the physical and chemical properties of quantum dots. Conducting the research on the endocytosis pathway of quantum dots in different cancer cells could provide scientific basis and clues for the treatment of cancer diseases.